Welcome to this series of hematopoiesis tutorials, and today we want to talk about erythropoiesis. If you haven't watched the videos on introduction part 1 and introduction part 2 on hematopoiesis, please go back and first watch them so that you can understand this. So today we want to focus on how red blood cells, what we call erythrocytes, are produced in the body. And this erythropoiesis, which is the formation of erythrocytes, because erythro comes from the word erythrocyte, whereas poiesis means formation. So we are looking at the formation of red blood cells. This is what we are focusing on. We are looking at formation of red blood cells in the body, whereby these red blood cells in the body, they are triggered by hypoxia. When does the common myeloid know that it should produce red cells? The trigger factor is hypoxia, whereby hypoxia is lack of oxygen. Lack of oxygen. That when the body is lacking enough oxygen in the tissues, we see the kidney. This one, hypoxia, triggers the kidney, the juxtaglomerular apparatus of the kidney to release erythropoietin hormone. And this erythropoietin hormone is the one that makes the myeloid stem cell to become committed to produce red cells. And this erythropoietin, it commits it to produce the burst-forming unit, erythroid, which gives rise to colony-forming unit, erythroid. And is the one that gives rise to the different stages we want to see. Whereby stage number 1 is always pronormoblast, which is the first to be recognizable, pronormoblast. Then it gives rise to the basophilic normoblast. Then it gives rise to polychromatic normoblast. Then this gives rise to orthochromatic normoblast. Then gives rise to reticulocyte, and finally the mature red cell, as we are going to see. So these are the only 1, 2, 3, 4, 5, 6 stages that are involved in erythropoiesis. So we want to describe these 5 different stages. And whereby pronormoblast, you can call it rubiblast. Another name you can call it rubiblast. This one you can call it prorubilicite. This one you can call it rubilicite. And this one you can call it rubilicite. Then this one is reticulocyte, or you can call it, under Roman whisker stain, you can call it polychromatic cell, or you can under Roman whisker stain. Then this is the mature. So these are the different names which you are supposed to be knowing. Another naming we can call pronormoblast to be the early normoblast, this one to be intermediate normoblast, and this one being the late normoblast. That is another name we can call them. So we want to describe each stage, that when there is hypoxia, which is triggering the kidney to release erythropoietin hormone, which gives rise to the cells to begin maturing. And before we start describing these stages, we need to know the requirements. And the requirements, we have seen, one is hormones. We need hormones, e.g. erythropoietin hormone. We need We need nutrients like iron. We need iron. We need amino acids, proteins, that is amino acids, to form a globin chain. And we need also vitamin B12. Vitamin B12 and vitamin B6 plus folate. So these are the major requirements during erythropoiesis, whereby we need erythropoietin hormone, we need iron, minerals like iron, we need the proteins like which are made up of amino acids, and we need the vitamin and the folic acid. So when we have those ones into place, and we have hypoxia, then we begin to develop into these stages. And we say the pronormoblast is the first recognized stage. It is the first recognizable stage in the bone marrow. And as I begin this, we need to note this. One is that as these cells are maturing, the size is going to be decreasing. As the cell mature from pronormoblast to basophilic to polychromatic to orthochromatic and they mature, the size keeps decreasing. And the point number 2 to note is that as the cell matures, also the nucleus condenses. And then after condensing, it is extruded out, and at the end we don't have the nucleus. So we shall see the nuclear cytoplasmic ratio reducing. And point number 3 is that as the cell matures from pronormoblast to the mature one, the chromatin pattern keeps on becoming condensed or clamped. It begins when it is fine, it becomes condensed, more clamped, and until when the nucleus is removed. And those are the 3 things I want you to note. 1, as the cell matures, the size decreases. 2, as the cell matures, the nucleus also decreases and is extruded out. And 3, also the chromatin pattern keeps on condensing. Those are the 3 things you don't have to forget. And I don't need to cram the size. So long as I know the truth, I have to make sure that the size keeps on reducing. So the size of the pronormoblast, it is 12 to 20 micrometer in diameter. And the nuclear cytoplasmic ratio here, almost the whole cell is covered by the nucleus. So we see the nuclear cytoplasmic ratio being 5 to 1. And nucleoli, we have 1 to 2 nucleoli present in the pronormoblast. Then the chromatin pattern is fine. It has a fine chromatin pattern. And the cytoplasmic staining is purple, or it is basophilic. You can call it basophilic. It is deep or intense basophilic because of the presence of the DNA and RNA. Then the structure, that this is the biggest. And whereby the nucleus is covering almost the whole cell, and sometimes it is round. The nucleus is round, and the chromatin pattern is fine. It has fine chromatin pattern with 1 to 2 nucleoli. And this cytoplasm stains basophilic. This is the pronormoblast. After seeing the pronormoblast, or what we call rubiblast, which is the first recognizable stage, now we go to the basophilic normoblast, which you can call prorubicite. Its size, as I told you, the size keeps on reducing. This one is 12 to 17 micrometer in diameter. Then the nuclear cytoplasmic ratio is 3 to 1, because as I told you, as the cell matures, also the nucleus condenses. Here, no nucleoli present. The chromatin becomes less, clamped. It is beginning to become clamped. The chromatin pattern, it is condensing. It begins when it is thread-like, then it begins to thicken. Then the cytoplasmic staining is still basophilic, or you can call it purple, because there is DNA and RNA still present, so it is taking the methylene blue of the Roman whiskey. In the structure, when you're drawing, you make sure it reduces, it becomes smaller than this. Because the size keeps on reducing, and the nucleus is also condensing, meaning the cytoplasm is becoming bigger. Then the chromatin pattern condenses or becomes clamped. And the nucleus is round and no nucleoli present. So this is how we can describe basophilic normoblast. So this nucleus, and sometimes this nucleus is not at the center, it is eccentric. So you can see it doing like this. It is eccentric but with clamping chromatin pattern. From there we look at the polychromatic. Poly means many, is taking up different shades of color. And it takes up different shades of color because at this stage, that's where hemoglobin synthesis begins. So HB synthesis begins here. That's why there is presence of hemoglobin, which is taking up the eosine dye, together with RNA and DNA, which is taking up the methylene blue. And this cell measures 10 to 14 micrometer in diameter. I don't need to cram. I know the concept that the cell has to be reducing in size. Another one who says 10 to 15 is right. Then also we know that the nucleus is condensing. So here the nuclear to cytoplasmic ratio is 1:1. No nucleoli present, or we can say it is absent. Nucleoli absent. Here the chromatin pattern is, it becomes clamped. It's clamped, or it is becoming thick. It has condensed. Then the staining is polychromatic. What are we meaning by being polychromatic? It is taking off both methylene blue and eosine, so sometimes we can call it pink. It has different shades of eosine and methylene blue. So it is pink, or sometimes some books call it mauve. Then if we can draw it, it has to become smaller than this. It has to become smaller. And here, the nucleus has continued to condense. And the chromatin pattern becomes more clamped. That is how we can see. So the nucleus keeps on condensing, and also the cytoplasm increases. That is what we can talk about under polychromatic. So under orthochromatic, or what we call metarubricite, we want to see that this, we saw HB synthesis began at polychromatic normoblast stage, but at orthochromatic here we are going to extrude our nucleus, and the cell we say it is pycnotic, whereby the size of the orthochromatic stage is 10 to 12 micrometer in diameter. The nuclear to cytoplasmic ratio is 1:2, or it can be 1:3. Because here the nucleus has condensed further, that has become more smaller than the cytoplasm. Here the nucleoli is absent. Here it is more clumped. The chromatin pattern becomes more dense or more clumped. Then the staining of this cell is pink due to presence of hemoglobin. As we saw that hemoglobin began to be formed at the polychromatic stage, so it is staining pink. And if we can draw this cell, it has become smaller. So it is smaller compared to the upper one, whereby the nucleus is eccentric, it is near the periphery. And we told you that it has become more clumped and is near to be extruded. So here it is where it is extruded. The nucleus is at the near periphery, ready to be extruded, and this is, we call it pycnotic. So this is where we release the nucleus. That is why we call it orthochromatic stage, but is staining pink. The cytoplasmic staining is pink. That is the maturation. The size has to keep on reducing, even the size of the nucleus has to keep on reducing. Then when we go to the reticulocyte, these ones we call them reticulocyte when we stain with supravital stains. That is like neomethylene blue and brilliant cressyl blue. Then we call them polychromatic when we use Romano whiskey stains. And why are they reticulocytes? They stain deep, they stain purple with supravital due to presence of remnants of RNA. We have removed the nucleus, but the cell still has remnants of RNA which are taking up the methylene blue, and they are best demonstrated using supravital stains. The size is 8 to 10 micrometer in diameter. The nuclear cytoplasmic ratio is not applicable because we have removed the nucleus. This one not applicable because there is no more nucleus, and even chromatin pattern not applicable because no nucleus present there. Then we see the staining of the cytoplasm with the supravital stains deep, it stains blue, or you can call it, it stains bluish with supravital stains, then polychromatic with Romano whiskey stains, due to remnants of RNA. So this is blue with supravital. Then polychromatic with Romano whiskey stains. Then the size has to reduce further. The size has reduced further, but is containing remnants of RNA which are staining blue or polychromatic. It lacks the nucleus because the nucleus was extruded at orthochromatic. So this is what we can talk about under reticulocytes. And these reticulocytes, they are the first, this is the first stage to be recognized in periphery. First stage, first immature stage. Mature stage seen in blood saturation. Don't forget this. They can ask you in objectives that which of the following stage, immature stage, is first seen in the blood saturation. And it should not always exceed, should be less than 1% in blood saturation. So don't forget that. It can be a multiple choice question. Then lastly, we want to look at a mature red cell. This mature red cell, it measures 6 to 8 micrometer in diameter. Of course, there is no nucleus, no nucleoli, because no nucleus, even here, no nucleus. It stains pink. Here it has become intense pink due to much hemoglobin has been formed. And this red cell, when I draw it, allow me to draw it here because of the space. This red cell, this mature RBC, the size also keeps on reducing. And the good news that it has area of central pera. This is not the nucleus. This is area of central pera, then surrounded by the hemoglobinized area. And this area of central pera, this one we are calling it area of central pera, which should measure a third of the cell, and then surrounded by hemoglobinized area. So this is hemoglobinized area, and we have seen it is measuring 6 to 8. And when seen on the top, it is always seen like a biconcave disc in shape. It's seen as a biconcave disc in shape, and this red cell is made up of spectrin and ankyrin, which gives it its flexibility during blood saturation. It is always flexible and is protected by cytoskeleton known as ankyrin and spectrin. And it is always surrounded by a negative charge, which creates the zeta potential from them not being together. And finally, the function of this red cell, we see it majorly to transport. It contains hemoglobin, which transports oxygen and carbon dioxide. Transports oxygen from the lungs to respiring cells, then transports carbon dioxide from respiring tissues or cells to lungs for exhalation. Then also the hemoglobin which it has, it acts as a buffer. This buffer meaning it maintains the body pH. So these are the three major functions of red cells. Transportation of respiratory gases, that is oxygen and carbon dioxide, then hemoglobin in the red cell acting as a body buffer to maintain the pH of blood. Thank you so much for listening from the start until the end, whereby we have seen things we have noted. We have said as the cell matures, size decreases. As the cell matures, the nucleus also condenses or becomes smaller, then it is extruded at the orthochromatic. And we saw as this cell maturing, the chromatin pattern also becomes dense. And we have seen three important things which they can ask in multiple choice question. That which of the following immature cell is first seen in the bone marrow? That is pronormoblast. Then the last the immature stage to be seen in the blood saturation is reticulocyte. Then HB synthesis begins at polychromatic stage. And even cell division stops at polychromatic stage. Beyond the polychromatic stage, there is no more cell division. There is only maturation. So proliferation stops here. We see also cell division stops at polychromatic. So they can ask you that which of the following stage during erythropoiesis does cell division stop? Stops at polychromatic, and HB synthesis also starts at polychromatic stage. And whenever you are drawing, make sure the size keeps on decreasing. That your drawings be in line with the words you're speaking, as the nucleus also condenses. Thank you so much. Always at this channel. Thank you for being with us. Always please share your comments and like the video if you have understood. Thank you so much.